Not necessarily, it's just kind of give you a reference. And what I want you to do is we're going to be working in groups of 3 for this whole session. So I want you to get yourselves clustered around in groups of 3, and we're going to work on these worksheets. So today this is a class in virology, so this is an upper level, mostly juniors and seniors, occasionally a few graduate students will come in here. And right now we're in a theme, we're talking about Ebola, and then we want to get into the idea of tracking. So how do we know where did the outbreak come from? And so we're going to do an activity, we're going to be working with some genetic sequences, and so they're going to be working in groups trying to line up sequences and see how we match things up and try and make connections between DNA sequences to understand the process by which we can use genetics to trace epidemics. So let's hear how you group things together. So your 2 groups back there kind of came up with the same thing. I guess it's something about that part of the room.
We saw that they tended to only have a C mutation and a T mutation. Sequence 14 had an extra mutation for A.
Created one by itself.
One that just couldn't fit in with anything.
I guess we grouped it a little bit differently. We just had the first C and T mutations.
But really the idea of what we learned in earlier classes, in my microbiology class, we talk about how sequence data comes about, how we use sequences to look at the evolution of organisms. And so we do a lot of activities there in that concept. And so now this is taking it to another level where they're going to have smaller pieces of sequence and actually doing that actual alignment part by themselves. In other classes they get kind of the computer thing. The computer does all the alignment for you, doing all those kinds of things. In this class, they're actually going to get the sequences themselves. They can line them up. They can see why doesn't it really work out that way. But the main objective is then how to use these sequences.
You can kind of sequentially see where the virus traveled to. Like if the group 2 ones were, west of the group 1 ones, and try to predict where it might spread.
This information and then take that to the treatment level. All right, thoughts, questions, comments? When I usually get end of course feedbacks, usually the things that are most pointed out are doing some of the active learning things. But the vast majority of students are very much in favor of doing something where they're working in groups and not sitting in a lecture. So they're actually actively doing something, either trying to figure out a problem, trying to solve a problem, trying to understand the basic data that's involved. So I think it's been a very positive response to these.